So today I am starting with the concept of the gene. Yesterday we have seen in brief what are genes, how the genes were termed. So today we are going to see in detail the concept of the gene. So gene, earlier the concept or the gene term was not present. In 1865, Gregor Mendel, when he carried out his experiment on the pea plant, he knew that specific characters are usually expressed in the individual peas. So what were those specific characters or the specific traits, like the color of the pods, then the tallness and the shortness of the plant, as well as the bean structure, etc. But these factors, they were called as the Mendelian factors, and they thought that this might be responsible for the functioning of the gene. Later on, the gene usually was termed in order to, this is particularly the gene is a Greek word which means to become or to grow, and usually it is made up of the DNA, and the DNA, as you know, it is made up of the nucleotide sequences or the nucleotides. You know that most of the prokaryotic as well as the all the eukaryotic organisms, they have DNA as their hereditary material. But few viruses like plant viruses, then reovirus and wound tumor virus, they have RNA as the genetic material. Means the genes or the hereditary material, it can be a DNA or it can be a RNA. Now about the history of the gene. Gene was first coined by W. Johansen in 1909, and later on in 1908, Garrod also proposed a theory of gene, and he said that one gene is responsible for synthesizing one product, and therefore the hypothesis of one gene one product came into existence. But after this, the research went into a slag period, and no research was carried out on the particularly the concept of the gene. Later on in 1911, gene theory was put forth by T.H. Morgan, but the exact means or the concept of the gene or the theory of the gene came into existence only in 1949 by L. Pauling and Ingram, and they established the role of genes in the protein synthesis. So Biddle and Tatum in 1948, then they proposed one gene one enzyme hypothesis. Firstly, it was said that one gene is responsible for making one product, but at that time they were not knowing what type of product was made. They called it as a expression of a particular phenotypic character. They were not knowing a particular enzyme is being synthesized or a particular protein is being made. The fine structure of gene was then proposed by Seymour Benzier in 1962, and then then the scientists came to know what the genes were made up of, how are they responsible for making an enzyme or a protein molecule. Then Janowski in 1965, he proposed one gene one polypeptide hypothesis, means that is one gene is responsible for making one polypeptide, and therefore one gene is usually making or synthesizing a polypeptide molecule or a protein molecule. Then the gene concept in detail was given by Sutton and Boveri in 1902. So this is a brief history or the evolution of the gene concept. In 1866, Gregor Mendel, he said that a unit factor that controls the specific phenotypic trait can be referred as a gene. Then later on in 1908 and 9, he said that one gene is responsible for one product or one metabolic block theory. Then Biddle and Tatum in 1940 one gene, one enzyme theory came into existence, and then in 1957 Ingram gave one gene, one polypeptide theory, and in 1960s C. Janowski and his co-workers gave that gene is a unit of recombination. It was then only when Shapiro and his co-workers in 1962 gave a complete structure of lac operon, and then they found out that gene is consisting of structural genes in the operon which can be forming polypeptide molecules responsible for making a particular enzymes. So, the gene definition to different scientists were different, but the gene to the geneticist is very similar to the atom for the chemistry, and the gene is the unit of genetic information which controls the specific aspect of the phenotype. So, gene is usually a unit of genetic information which specifies the function like making a enzyme molecule, a polypeptide molecule or a protein molecule. So, as we have seen earlier that this is a eukaryotic cell, typical eukaryotic cell in which nucleus is present, and nucleus when it is extended it consists of a DNA molecule, that the nucleus is consisting chromosomes. You know that different types of chromosomes are present in prokaryotic as well as the eukaryotic cell, and then if you see the extended structure of DNA then undergoes a transcription process and translation to form a protein molecule. So, the genes are the fractions of or a part of DNA molecule which is regarded as the genetic material. Now, T.M. Morgan proposed the gene theory. So, until now we were knowing only the concept of the gene, but the gene theory was put forth by T.M. Morgan. What did he say? He says that the chromosomes, they are the bearers of hereditary units because they carry the hereditary or the genetic material from parent to the offspring, and they carry all the phenotypic traits, all the particular genes from parent to the offsprings. And how it is carried out? It is carried in the form of a chromosomes, because chromosome carries hundreds or thousands of genes. And how the genes are arranged? The genes are arranged on the chromosomes in a linear order, and they have a special regions where they are located. That particular region present on the chromosome is called as the locus. So, this figure depicts the structure of the cell. Here you have a nucleus, then chromosomes are located, and then this is the eukaryotic chromosome, therefore you have a nucleosome units. So, the genes, they are located. So, in 1928 Belling proposed that the strings on the beads, so this is the string of the DNA on the bead is the histone protein, they carry the genes, and these genes were responsible for making a particular enzyme or a protein molecule. So, it was given by Belling in 1928. Then genes are both basic functional units, and they are the smallest genetic structural units. So, in 1940 the gene was considered as a basic unit of genetic information, and therefore, the unit of function or controlling the inheritance of one character or which attributes a phenotypic expression is called as a unit of function, and therefore, the gene was considered only as a basic unit of genetic information and as a unit of function and as a unit of structure. So, the unit of structure means operationally it is defined by recombination and a mutation. So, what are the different terms which are related to the gene? That is, we have gene acting as a unit of function, which we call it as cistron. The genes, when it is acting as a unit of recombination, then we call it as a recon, and when it is acting as a unit of undergoing mutation, then we call it as muton. As well as the gene can be a unit of undergoing a complementation, which you call it as a Complon. So, you have two different theories or definitions of gene. So classical definition of gene and a modern definition or the molecular definition of genes. So first we will see the classical definition of genes. So gene as a unit of function, it means that one gene is specifying one character. Here it is said that the Mendelian factors is responsible for a particular phenotypic trait, and therefore it was in the classical definition of gene, it was considered that gene is a undivisible unit. It is considered as a undivisible, means the gene cannot be cut, means that particular gene which is carrying all the hereditary information cannot be cut into, or it is not having a recombination gene, or it is not having any mutation sites or recombination sites. So it was only considered that it is specifying one phenotypic character. But it was introduced in Saturn 1902, and then it was elaborated by Morgan, Beige and Mueller, and then they said that genes are the discrete particles inherited in Mendelian fashion that occupies a definite locus in chromosome and is responsible for expression of a specific phenotypic character. So it was again elaborated by this particular 3-4 scientists, and the definition of classical concept of gene was put forth. So number of genes in each organism. Now what happens when the Mendel carried out his experiment on the pea plant? Usually he studied the phenotypic characters only, and the scientists came to know that these phenotypic characters are many more when compared to the number of chromosomes in a plant or in a whatever the test organism they used. So the number of genes usually in an organism is much more than the number of chromosomes, and therefore on the chromosome you have several number of genes located. The genes are arranged in a single linear order like beads on the string. As I have said, Belling in 1928 he said that the beads on the string carries genes, and these genes are responsible for one particular function. So these genes, they occupy a specific function on the chromosome which you call it as a locus, and if the position of the gene changes, then you know that the character or the phenotypic expression of that particular gene also differs or they alters. So gene can be transmitted from one parent to the offsprings. So genes may exist in several alternate forms which you call it as the alleles. Now genes are capable of combine together. They can combine together or they can be replicated during the cell division. So in order to pass on the genetic material from parent to the offspring, the genes usually they are duplicated or they undergo a replication process during the cell division cycle. Also, the genes undergo a sudden change in position and composition, and that particular phenomena was called as the mutation. So genes are capable of undergoing self duplication also to make their own exact copies, as we will see in the transposable elements or the transposons, or we call it as the jumping genes. So these genes are making their own copies and jumping from one location to the another one. So now we will see the modern definition of gene or the molecular definition of genes. So earlier it was said that gene is usually carrying the genetic information and is responsible for a specific trait of phenotypic expression or phenotypic trait. It means that that particular gene was undivisible, it was not having any recombinational sites, or it was not having any mutations. But later on the modern definition was put forth, and it is said that gene is the unit of genetic information, the sequence of DNA that specifies one polypeptide. Means they have come to the conclusion that it is not only a phenotypic expression, it is the gene which is making a one polypeptide molecule. So it includes coding as well as, now since we know that the polypeptide is first made via translation process, but before the translation process it has to undergo a transcription, and transcription is nothing but a synthesis of messenger RNA. So, in eukaryotes, while doing the messenger RNA synthesis, you have both introns and exons. Introns are non-coding regions and exons are coding regions. So, usually the non-coding regions or the introns or intervening sequence, they are removed out by the splicing mechanism, and the exons are joined together to make a mature messenger RNA molecule, and then this messenger RNA molecule is responsible for making the polypeptide chain. So, Benzer in 1957, then he defined he gave a different terms for different nature of genes and genetic material in relation to the chromosome on the basis of genetic phenomena to which they involve. So, accordingly, he named in 1957, he named genes as a unit of transmission, that is the cistron, or in simple terms as a unit of function. Then genes as a unit of recombination, recon, and genes as a unit of mutation or the mutons. So, as you see, observe here in this diagram, that this is your chromosome, and on this chromosome these are the particular genes, this gene 1, gene 2, gene 3, gene 4 and so on. So, in one particular chromosome you can have thousands of genes located at specific location, and that is what is called as the locus. So, now we will come to the point, that is genes as a unit of transmission or cistron. So, the part of the DNA which specifies a single polypeptide chain is termed as cistron. Cistron can have 100 nucleotide pairs in the smaller or you can say not very complex microorganisms like unicellular organisms, and also to these nucleotide pairs, it can be up to 30,000 nucleotide pairs in the eukaryotic organisms. So, one cistron or one gene can contain 100 nucleotide pairs in length to 30,000 nucleotide pairs as in humans. What it does, it transmits characters from one generation to the another as a unit of transmission. So, this is the smallest segment of DNA capable of being separated and undergoing the exchange with chromosome, which is called as a recon, and therefore, the gene is considered as a unit of recombination or recon. So, as a unit of recombination means when the DNA is in homology or during the cell division, in mitotic division, you will see that the two pairs of chromosomes, that is particularly the alleles, come together and they undergo a genetic exchange. And when this genetic exchange takes place, so that is the genes during the segregation process, and then when they segregate out, the two daughter cells will have different genes on their alleles. So, a recon consists not more than two pairs of nucleotides. So, two pairs of nucleotide is more than sufficient to undergo a recombinational event. Then genes as a unit of mutation or muton. So, muton what? Muton is a smallest division or a smallest subunit of genetic material which when changed or mutated produce a different phenotypic trait than the original one. So, this muton, it can be a single nucleotide also, as we have seen the example of sickle cell anemia, in which only the one that is the glutamic acid, if it is exchanged with the valine, then usually what happens, the structure of a globular heme protein changes to the sickle shaped and it results in a sickle cell anemia disease. So, this is how the mutation takes place. So, now we will see that when you consider hereditary material, the total hereditary material it is found in the nucleus, but outside the nucleus also you have certain organelles, and these organelles also carry their own DNA. So, the total hereditary material can be divided into genome and plasmon. Now, what is genome? Genome is the hereditary material of the chromosomal complex, and plasmon is the hereditary material outside the chromosome. Got this point clear? When the hereditary material is present or the genetic information is carried in the nucleus or on the chromosomes, then you call it as the genome. But plasmon means it is the hereditary material outside the chromosome. So, the plasmon term was introduced by Fritz von Wettstein in 1924. Then as you know that when you call it as a plasmon, they have their own genes, and this gene is referred as or termed as plasma gene, which was coined by Darlington in 1939. So, what are those plasma genes? How they are named after? They are named after in which location or in which cell structures you observe those plasma genes. Accordingly, they have been named as. So, the plasma genes when they are present in the plastids of a plant cell, then you call it as a plastogenes. When it is present in the kinetosomes, they are called as a kinetogenes, and when they are present in mitochondria, then they are called as chondriogenes, and when it is present in the centrioles, then it is called as the centriogenes. So, according to the location at which cell structure it is found or in which cell structure it is located, accordingly these plasma genes, they are called by different names as plastogenes in plastids, kinetogenes in kinetosomes, chondriogenes in mitochondria, and centriogenes in centrioles. So, extensive work has been carried out by the molecular biologist, and the nature of gene has now become very crystal clear, and because of this the gene is now defined as a polynucleotide chain with the discovery of 3D or three-dimensional structure by Watson and Crick DNA structure, and also the DNA as a genetic material by contributions made by different scientists like Griffith's experiment in 1928, then Avery, MacLeod and McCarthy's experiment. It laid the foundation that DNA is the hereditary material in prokaryotes, that is particularly the bacteria, and in eukaryotic organism. So, the DNA is now consisting of fractions or segments, and that is referred as a gene. So, gene is also a polynucleotide chain, and it consists of segments, each which is controlling a particular trait. Now, accordingly, the genes are considered as a unit of function, a unit of mutation, and a unit of recombination, which we are going to study in detail. So, gene as a unit of function or the cistron. Now, first link between gene and gene product was established in 1909 by E. A. Garrod when he was working on a gene product which he called it as protein. So, he defined or he gave the hypothesis one gene, one protein. But until 1940 his work was ignored, and later on in 1941 Biddle and Tatum put the theory or the concept of gene as one gene, one protein hypothesis. Then later on encouraged by this link between the gene and the protein, so they came to know a relation in between the gene and the protein molecule, and because of this many researchers now they got interested, and then they investigated or they carried out research on the mechanism by which DNA is forming a protein molecule. So, how the DNA was translated into the structure of protein. So, soon it became clear that DNA is first transcribed into the RNA, which you call it as a messenger RNA, and then which is subsequently the messenger RNA information is translated into a protein molecule. So, in 1908 Garrod as well as some of the investigators suggested that genes produce proteins which act as an enzyme. So, Garrod, he said that one gene is responsible for making a protein that can act as an enzyme. So, he gave the hypothesis, that is one gene, one enzyme hypothesis. But later on, enzyme is not always produced. It can be a structural proteins also. It can be another subunits of enzyme molecule. So, it is usually a that is the polypeptide molecule. So, gene represents a particular sequence of nucleotides along DNA molecule which acts as a unit of inheritance. So, this particular unit of inheritance is called as a cistron. Since gene forms a messenger RNA molecule, it is said to be one gene, one messenger RNA, but in prokaryotes you know that several genes form a single messenger RNA because they are in continuous, or we call it as a contiguous genes located, and therefore that contiguous gene is forming a messenger RNA, and therefore this messenger RNA is called as a polycystronic gene because you have many genes. As we have seen in lac operon, you have three genes, lac Z, lac Y and lac A. So, these three genes are in continuous which are transcribed together at a one go to form a single messenger RNA molecule, and then this single messenger RNA molecule can give you three different enzyme molecules like beta galactosidase, transacetylase and permease enzyme. So, this is a polycystronic molecule. So, the gene is a polycystronic gene. Therefore, this concept was later on again formulated, and it was called as one gene, one polypeptide hypothesis. So, since an enzyme or a protein may contain one or more polypeptides, it was very clear that one should give the hypothesis that one gene is responsible for making one polypeptide molecule. So, Stahl had defined gene as a polynucleotide sequence of DNA controlling expression of particular trait. So, gene as a unit of function, what it represents? It represents the segment of DNA molecule and consists of linear sequence of nucleotides which controls some cellular function. So, when you observe a particular E. coli cell has a cistron which contains near about 1500, that is 1500 base pairs, but in some others it may be as many as 30,000 nucleotides in a single gene. So, each cistron is responsible for coding one messenger RNA molecule, and which in turn codes for a polypeptide chain, and this polypeptide chain can be a enzyme or a protein molecule. So, the cistron may have hundreds of units of mutation. So, in the cistron you can have units of mutation as well as you can have units of recombination. Means within the gene of a cistron or a functional gene, you can have a recombinational gene also, you can have a mutation gene also. So, cistron occupy much greater area in the chromosome when compared to the mutons and the recons. So, this cis and trans arrangement of alleles, so this is a eukaryotic allele. When do you call it as a cis genes? When they are located side by side, then you call it. A is one gene and the B is the another gene. When it is located on the same allele side by side, then you call it as the cis gene. So, usually it is found in the wild type. But what happens during the recombinational event? Usually the allele can undergo a exchange of genetic material in between the two alleles, and therefore what happens, A gene will be located on the another allele and B gene will be located on the another allele. So, that type of usually arrangement is called as gene arrangement, is called as the trans arrangement of alleles, and this is a mutant one. So, if you consider a gene, this gene is A, B, C, D and so on, it is making a polypeptide gene respectively A, B, C, D. Okay. So, how will you understand it is a cistron, it is a recon and it is a muton? So, this gene when it is making its duplicate copy, which is represented as A dash, then B, B dash, C, C dash. So, it is a duplicated copy of this particular genes. So this is your parent one, and this is your which is after duplication, which is going to be segregated in the daughter cell. So this is your one complete chromosomal sequence which has many number of genes. And how recombination usually is done? So during the duplication, you will see that the recombination is taking place, because there is a homology in between these two. So even if there is a change in the two nucleotides or two nucleotide bases, then you will see that the chain has been resolved. So this has been cut off and this has added here, and here it has been added to this chromosome. So the recombination has taken place at this level. And muton means when something when a nucleotide has undergone a mutation. So earlier it was having, this particular gene was having A, but now that A had got removed out, and instead of A now T has got inserted, or G has got inserted, or C has got, means the normal T now is replaced by either a C, T, normal A has been replaced by C, T or G, and therefore it has resulted in the mutation. So this is what is called as the mutation. So gene as a unit of recombination, so or you call it as a recon. Earlier it was thought that crossing over occurs between the two genes. So how it was given, how the experimental evidence was given? In 1962, Benzer demonstrated that crossing over or recombination occurs within a functional gene or the cistron. Means cistron itself is undergoing a recombinational event. So in a cistron, the recombinational units may be more than one. So in a big recombination, that is cistron molecule or a functioning gene molecule, you can have as many as recombinational units. So the smallest unit capable of undergoing recombination is called as a recon. So Benzer found that when the cultures of T4 bacteriophage, they were growing on the agar plates of host Escherichia coli, the normally T4 formed small and smooth edged plaques. Now whereas the mutant T4 phages, they form larger plaques, and these plaques were rough edged. I will show you in the figure. So normally this is so because that the mutation has occurred. Where the mutation has occurred, the mutation has occurred in the R2 region. So formation of rough edged plaques, it was governed by two adjacent genes, means the cistron R2A and cistron R2B. So in cistron R2A, you have as many as that is mutation sites. So both these regions, they function independently. R2A and R2B, they function independently, and it is consisting of 2500 and 1500 nucleotides respectively. So this R2A is having 500 mutational sites where you can observe the crossing over may occur. So when the crossing over takes place, exchange of two segments takes place, and because of this, it results in the formation of a mutant plaque, which is rough edged plaques and normally larger in size when you compare with the wild type phage, which produce a smaller plaques and a smooth edged plaques. So this is the diagram. Here you will see that the plaques are very clear, which is smooth plaques. So usually these plaques, they are a smooth one, but this one usually it is when it is undergoing a mutation, those rough plaques, they are very large. See here, and it is a rough, it is not a smooth. Smooth means the entire edge is a round one, and when you observe the mutant one, the mutant one is having a rough edge, means it is usually like so. If you observe, it is a rough plaque and it is larger in size, and similar results were obtained by Benzer, and thus the work of Benzer led or he supported that the gene is undergoing a recombinational event, and therefore there is a recon which is located in the functional gene, you call it as a cistron. So this is the site of R2. In the R2 in the R2 region of cistron, you have two different cistrons, that is R2A and R2B, and in the R2A region you have 500 mutational sites. So 100 pairs of nucleotides is responsible for undergoing 500 mutational sites or mutations can occur. So this is how, this is your R2 region. So here it is representing blue, here it is representing it. When it is undergoing a crossing over, the blue has come here, so you are having a mutation and this, and whereas you don't have any mutation, and therefore this is your wild type and this is your mutant one. So wild type is producing the smooth plaques, but this one which has got mutated now, it will have a larger and turbid, or that is the rough plaques. So genes as a unit of mutation or muton. So Benzer in 1962, he also coined the term muton to denote the smallest unit of chromosome which undergoes a mutational change. So muton may be defined as the smallest unit of DNA which may be changed, and that change is nothing but due to the nucleotide change. So if a single nucleotide is undergoing a change, then it can result in the mutation. So cistron is the largest unit in size, followed by the recon and the muton. So this can be explained as the gene consists of several cistrons, and this cistron also contains a recon, and recon contains a number of mutons. So in a cistron gene you have a recon, and in a recon gene you can have a muton. So if the size of a recon is equal to the muton, then there would be no possibility in recon for consisting of several mutons. So usually what happens, if the size of muton is more or equal to recon, then there is no possibility that recon is consisting the several, because it will be a equal one. So undergoing a recombinational event or undergoing a mutational events, it would be the same one. So thank you. I hope you have understood the lecture. If you have any questions, you can ask me. I am now opening the discussion forum.